Welcome to foodfeedinfo.com. This is part 3 of Carbohydrates Metabolism. I will discuss Pentose Phosphate Pathway of Glucose Oxidation. This pathway is also known as Hexose Mono-Phosphate shunt, and it is a direct oxidative pathway. This is also known as Dicken-Horecker pathway. If we look at the animal tissue, in animal tissues, the major catabolic fate of glucose 6 phosphate is glycolytic breakdown to pyruvate. This pyruvate is then converted into acetyl CoA and oxidized via TCA or citric acid cycle. And that results in the production of energy in the form of ATP. The glucose 6 phosphate have many other catabolic fates that lead to the production of some specialized product. And pentose phosphate pathway is also an alternate pathway of glucose oxidation. Here, oxidation of glucose 6 phosphate is into pentose phosphate or 5 carbon carbohydrates. In humans, about 10% glucose per day enters this pathway, and about 30% glucose in red blood cells and liver is metabolized by this pathway. In this oxidative pathway, NADP is the electron acceptor and it yields NADPH. Particularly the rapidly dividing cells like bone marrow, intestinal mucosa, skin, they uses this pentose to make deoxyribonucleic acid DNA, RNA, and such coenzymes as ATP, NADH, FADH2, and coenzyme A. However, in other tissues, the essential product of this pentose phosphate pathway is not the pentose but the electron donor NADPH. This electron donor is required for reductive biosynthesis or to counter the damaging effect of oxygen radicals. The tissues that carry out extensive fatty acid synthesis, like mammary gland, lactating mammary glands, adipose tissues, liver, or very active synthesis of cholesterol and steroid hormones like liver, adrenal glands, and gonads, they require the NADPH provided by the pathway. Erythrocytes and the cells of the lens and cornea, they are directly exposed to oxygen, and thus they are prone to damaging effect of free radicals then generated by oxygen. So if we consider the importance of pentose phosphate pathway, NADPH or electron or hydrogen donor production is the major role of this pathway, because this NADPH is needed for glutathione reduction, reductive biosynthesis, which is required to counter the damaging effect of free radicals. So in simple way, we can say it is required for the detoxification of free radicals, particularly in RBCs. Uh, NADPH is also required for keeping iron of hemoglobin in ferrous form, and it also keeps many enzymes like thiol containing enzymes in their forms. The cells or tissues which are doing this fatty acid synthesis, cholesterol, and steroid synthesis, they also require NADPH. So if you look at this figure, you can see that this glutathione, it get reduced uh, by NADPH, and this reduction is caused by glutathione reductase. This reduced glutathione then detoxify the free radicals or oxidizing agents like hydrogen peroxide and the drugs which are responsible for this oxidative damage. So it get oxidized again into glutathione, which then again converted into reduced glutathione if NADPH is there, and glutathione reductase enzyme is catalyzing the reaction. So briefly, here you can see there is a oxygen demand in very active cells in the mitochondrial respiration, ionizing radiation, sulfur drugs, herbicides, anti-malarial drugs, and divisine. So either due to mitochondrial respiration or other drugs, this oxygen is converted into superoxide radical, and it takes an electron and then converted into hydrogen peroxide, which have a very damaging effect. This is an oxidizing agent, and when it takes another electron, it is converted into free radical, hydroxyl free radical. So it causes oxidative damage to lipid, proteins, and DNA. So if there is no control mechanism or countering mechanism for these free radicals, the cell get destroyed. Now look at this. There is an inhibitory sign or cross sign, hydroxyl free radical and their oxidative damage. And this control is due to the presence of reduced glutathione. It protects the cell by destroying hydrogen peroxide as well as hydroxyl free radicals. And regeneration of this glutathione from its oxidized form, it requires NADPH, that is produced in the glucose 6 phosphate dehydrogenase reaction. So now look at the complete picture. Oxygen is converted into hydrogen peroxide, and glutathione peroxidase is converted this hydrogen peroxide into water molecule. If there is a presence of reduced glutathione 2GSH, that is converted into oxidized form. And then again, if you look at the cross, the hydroxyl free radicals, they are not going for damaging effect, rather they are countered by glutathione, reduced glutathione 2GSH. And this 2GSH is converted into GSSG, reduce from to oxidized glutathione. Uh, then again, this oxidized form is converted into reduced form, and that require an enzyme, glutathione reductase, from where they are getting this hydrogen ion, and that is coming from NADPH. So NADPH is converted into NADP with the enzyme that catalyzes this reaction, glutathione reductase. And if you look at the lower part of this slide, the glucose 6 phosphate is converted into 6 phosphogluconolactone. So this conversion is due to the presence of an enzyme, glucose 6 phosphate dehydrogenase. So this enzyme convert glucose 6 phosphate into 6 phosphogluconolactone. During this conversion, there is a production of reduced NADPH from NADP. So this is a major, you can say, the reaction, okay, that is responsible for the production of NADPH, that then cause a reduction of the glutathione, and that reduced glutathione then counter the free radicals. Then comes other importance of this pentose phosphate pathway, that results in the production of ribose sugar, and this ribose is required for the synthesis of RNA and DNA. Particularly the rapidly dividing cell, they require this pentose sugar for RNA and DNA synthesis. Pentose phosphate pathway, or HMP shunt, it occurs in the cytosol of all cells, and it has two phases. The one is oxidative phase, the other is non-oxidative phase. The oxidative phase is irreversible, and it generates NADPH, and there is a removal of carbon dioxide. During the non-oxidative phase, there is a production of ribose phosphate and glucose 6 phosphate. However, this reaction is reversible when we compare it with the oxidative phase. Now we look at the first step is the conversion of glucose 6 phosphate into 6 phosphogluconolactone. This reaction is catalyzed by glucose 6 phosphate dehydrogenase. During this step, NADPH reduced into NADPH. And in the next step, this 6-phosphogluconolactone is converted into phosphogluconate. The phosphogluconate is then converted into a 5-carbon ribulose. This reaction is catalyzed by 6-phosphogluconate dehydrogenase. During this reaction, again NADP is reduced into NADPH, and there is also a release of carbon dioxide. So this is the oxidative phase of pentose phosphate, where glucose 6 phosphate is converted into ribulose 5 phosphate with the production of 2 NADPH. Now about non-oxidative phase. During non-oxidative phase, the pentose phosphates recycle to glucose 6 phosphate. The tissue which require primarily NADPH, the pentose phosphate produced in the oxidative phase of pathway are recycled into glucose 6 phosphate. In this non-oxidative phase, ribulose 5 phosphate is first epimerized to xylulose 5 phosphate. So in the next step of non-oxidative phase, xylulose 5 phosphate is converted into glyceraldehyde 3 phosphate by transketolase enzyme. Next, glyceraldehyde 3 phosphate is converted into fructose 6 phosphate by transaldolase enzyme. Fructose 6 phosphate is further utilized through the glycolysis for the production of energy, or it can be converted back into glucose 6 phosphate. Now look at on the right side of the reactions. Ribulose 5 phosphate is converted into xylulose 5 phosphate by epimerase, and this xylulose 5 phosphate is then converted into glyceraldehyde 3 phosphate by transketolase activity, and this 3 carbon glyceraldehyde is then utilized through glycolysis. Now in the center portion, that is basically running the right and left of the reactions, where through isomerase enzyme ribulose is converted into ribose 5 phosphate. Ribose 5 phosphate is then converted into pseudohaptolose and then into erythrose. Erythrose 4 carbon is then converted into fructose 6 carbon molecule, and that fructose is then converted back into glucose 6 phosphate. So during the non-oxidative phase of pentose phosphate pathway or HMP shunt, these reactions convert pentose phosphates to hexose phosphates that allow the oxidative reactions to remain in action or to remain continue. Most importantly, the enzymes transketolase and transaldolase, they are specific to this pathway. The other enzymes also serve in the glycolytic or gluconeogenic pathways. All enzymes in the pentose phosphate pathway are located in the cytosol, like those of glycolysis and most of those of gluconeogenesis. In fact, these three pathways are connected through several shared intermediates and enzymes. The glyceraldehyde 3-phosphate formed by the action of transketolase is readily converted to dihydroxyacetone phosphate by the glycolytic enzyme triose phosphate isomerase, and these two triose can be joined by the aldolase as in gluconeogenesis, forming fructose 1,6 bisphosphate. Alternatively, the triose phosphates can be oxidized to pyruvate by the glycolytic reaction. The fate of triosis is determined by the cell's relative need for pentose phosphates, NADPH, and ATP. Regulation of pentose phosphate pathway. The key enzyme in pentose phosphate pathway is glucose 6 phosphate dehydrogenase. NADP activates this enzyme while NADPH inhibits the enzyme. So the deficiency of glucose 6 phosphate dehydrogenase is an inherited sex-linked trait. Although the deficiency occurs in all the cells of the affected individual, but it is more severe in RBC. In RBCs, the hexose monophosphate shunt is the only mean of providing NADPH. So decreased activity of G6PD or glucose 6 phosphate dehydrogenase decreases the synthesis of NADPH in RBC. So this result in the storage of methemoglobin and peroxides in RBC, and this would lead to hemolysis. So as a result of deficiency of glucose 6 phosphate dehydrogenase, uh, the RBC wouldn't be able to carry the oxygen and they get destroyed. So this is a better presentation of this regulation mechanism of pentose phosphate pathway, where the key enzyme is glucose 6 phosphate dehydrogenase. This enzyme converts glucose 6 phosphate into pentose phosphates via the intermediate 6 phosphogluconolactone. And there is a production of NADPH, and a high concentration of NADPH actually inhibit the pentose phosphate pathway or the key enzyme which is converting glucose 6 phosphate into 6 phosphogluconolactone. Glucose 6 phosphate is partitioned between the two pathways. Either it can go to the glycolytic pathway or it can follow the pentose phosphate pathway. Whether glucose 6 phosphate enter glycolysis or pentose phosphate pathway, it depends upon the current need of the cell and on the concentration of NADP in the cytosol. So without this electron acceptor, the first reaction of the pentose phosphate pathway, which is catalyzed by G6PD, it cannot proceed. So when a cell is rapidly converting NADPH to NADP during the biosynthetic reduction, the level of NADP rises. So it allosterically stimulating G6PD and thereby increasing the flux of glucose 6 phosphate through the pentose phosphate pathway. However, when the demand for NADPH slows, the level of NADP drops, the pentose phosphate pathway slow, and glucose 6 phosphate is instead used to enter into the glycolytic pathway. So how this NADP and NADPH, they are regulating the pentose phosphate pathway. Here is the summary of overall reactions of pentose phosphate pathway, both oxidative and non-oxidative phase. During the oxidative phase, glucose 6 phosphate is converted into ribulose 5 phosphate, and that ribulose 5 phosphate is utilized for the synthesis of DNA and RNA, whereas through the activity of transketolase and transaldolase activity, this ribulose 5 phosphate again converted into glucose 6 phosphate, and this is the non-oxidative phase of pentose phosphate pathway. During this oxidative pathway, there is a production of NADPH. So NADP reduced into NADPH, and that NADPH again oxidized into NADP when there is a utilization of glutathione by glutathione reductase, and it causes reduction of glutathione into glutathione GSH. So NADPH formed in the oxidative phase is used to reduce glutathione GSSG and to support reductive biosynthesis. The other product of the oxidative phase is ribose 5 phosphate, which serve as a precursor for nucleotide coenzymes and nucleic acid. In the cells that are not using ribose 5 phosphate for biosynthesis, the non-oxidative phase recycles 6 molecules of the pentose into 5 molecules of hexose glucose 6 phosphate, and that allow the continuation of the production of NADPH and converting glucose 6 phosphate to carbon dioxide. So this is all about pentose phosphate pathway or hexose monophosphate shunt. I hope you have a deep insight and have a good knowledge of this pathway and would be able to understand the production of NADPH and their utilization in the body. So in the next lecture we will discuss glycogen metabolism. So that would be the part 4 of carbohydrate metabolism. So stay tuned with the www.foodfeedinfo.com, and thank you for your time and watching the video.